Hi, my name is Purna Trajan. I'm introducing Team Electrode's Junior Colloquium presentation. Presenting with me tonight is Katie Lacomi, Everett Lodarno, Megan Lynn, and our team name ELECTRODE stands for Evaluating Linear Radio Electrode Conformations for Tissue Repair and Organizing a Device for Experimentation. 1.9 million Americans in 2010 were diagnosed with diabetes, and 15% of these cases reported were also reported of having diabetic ulcers. Diabetic ulcers occur due to diabetic neuropathy. Diabetic neuropathy also stands for nerve damage. And these diabetic ulcers worsen due to poor blood circulation, lack of tissue repair. Diabetic ulcers tend to be the focus of our presentation, of our project. There are three main problems with current treatments for diabetic ulcers. The first one being that they require constant application from the patient. The second one is they don't directly target the wound. And the third being that they're expensive. Expensive not only in cost, but also in side effects, as the side effects range from allergic reactions to increase in necrotic tissue. Our body has a natural bioelectric healing system. This natural healing system occurs in the human epithelial tissue layer and consists of the transepithelial potential, transelectric potential. This potential ranges from 10 millivolts to 60 millivolts and causes the wound site to be positive relative to the surroundings. This induced electric field causes cells to migrate with the direction and results in angiogenesis. And our project will be looking at how electric stimulation through inducing angiogenesis can result in the wound healing process. Our primary hypothesis for this project is that electric stimulation affects the wound healing process through its effects on angiogenesis. We will be looking at three different factors for angiogenesis, two being the expression layers of VEGF and VFGF. VEGF is the vascular endothelial growth factor, and VFGF is basic fibroblast growth factor. And our third one is the one we discussed in our previous slide of cell migration through directionality through electric stimulation. We primarily hypothesize that through inducing these three factors indicative of angiogenesis, the electric stimulation will improve the natural wound healing process. Our three specific aims include: one, optimizing a linear applied electric field and testing its effects on inducing angiogenesis through its effects on cell proliferation, migration, VEGF, VFGF expression levels. And two, testing a radially applied electric field on angiogenesis factors, cell migration, VEGF expression, VFGF expression, and ultimately using these two data to find an optimized electric field and its effects on a diabetic rat model. Our first two specific aims of testing a linear electric field and a radially electric field and seeing its effects on the angiogenesis factors are done in vitro. And our in vitro experiments involve the rat aortic endothelial cells. These cells were used because they not directly affect diabetic ulcers, and they also express growth factors that are similar to cell proliferation and migration. Additionally, the in vitro rat models will also be looking at endothelial cells, so there will be a positive correlation when we move on to the in vivo model.
Thank you, Porna. I'm going to talk about the device. For our device, we are trying to use the best parameters we determined from our experiment to decide what to use for our final prototype. Our goal is to create a small and cheap prototype for our device to provide electrical stimulation. For our prototype here can be, our electrical setup can be modeled by this circuit with the cells and media representing by a electric resistance and the terminals of the battery representing the positive and negative electrodes. For our setup, we're going to be testing different voltages in two different electrode setups. On the left, we have our linear electrode setup, which is made up of two glass slides coated in titanium. The electric field between the two electrodes is relatively constant, and the cells will migrate from in the direction of the current, which is from the positive electrode to the negative electrode. In this case, to the right. On the right, we have our radio electrode setup, which is made of a titanium ring for our positive electrode and a rod in the center for our negative electrode. The electric field is radial and increases towards the center of the electrode. The cells will migrate towards the center of the electrode. And for the voltages we're testing for our in vitro test are 0.01 volts, 0.1 volts, and 1 volt, all at 50 hertz. We're going to be testing 3 of our 6 wells on our 6 well plates for about half an hour each. We're going to use a function generator to create a square wave for the voltage. The amplitude of the square wave is the voltage applied to the cells. And the square wave has a duty cycle of 50%, which means it's on half the time and off half the time. In here we have our linear setup simulation. In the right corner at the bottom, you can see our function generator that creates the square wave connected to our linear setup. And now Eddie will talk about cell analysis.
Thank you, Kate. So, in order to answer the previously mentioned specific aims, we are using 4 different experimental aspects. The first one is testing for cell proliferation and cell viability. The second one is testing for angiogenic growth factor expression, and the third one is looking at cell migration. So for our cell viability and cell proliferation assays, we're using something called a ELECTRODE assay, which is very simple. It consists of fluorescently staining our cell samples in each one of our wells and exposing them to a fluorescent light. So after we do this, we examine our samples under a fluorescent microscope, we take pictures, and then we analyze these pictures to get a cell density count using computer software in each day. So the reason why we are calculating cell densities instead of an absolute cell count is because first, calculating an absolute cell count can be difficult and it's not very reliable. And second of all, by using cell densities, we can normalize our data and we can use it for comparison. So far, our studies show that there's no significant cell death after we have achieved a successful cell culture. And this is a picture of one of our sample live dead pictures. And the green cells, the green dots represent live cells. And if any, any red dots will obviously correspond to dead cells. So as you can tell, so far, most of our studies show that electrical stimulation is indeed not killing our cells. So as Borna previously mentioned, angiogenesis is directly correlated to an increase in cell proliferation. So in order to do that, I'm going to show you one of our preliminary data that we have gathered. This shows the cell density counts for 24 hours and 48 hours after testing with 0.1 volts on our cells. And as you can tell, in general, we have a higher cell density count in our experimental, which are our stimulated cells, compared to the control cells, which are non-stimulated. And also we see a higher cell density count in 48 hours post-stimulation than 24 hours after. Now, this little chart represents the progression of our experimental assays. The yellow shaded items are the assays that we have accomplished so far. So as you can tell, we have done live data on our first specific aim. And we have done mRNA extraction from our cell samples. So this extracted mRNA is it's tested using something called a nanojob. So we're looking at the absolute quantities of mRNA in our samples and also testing for its purity. And we are taking these samples and freezing them for further testing. So the blue shaded items are the things that are the next step on our experimental assays. And first we are testing for gene expression of VEGF and DFGF. And in order to do this, we are going to be doing something called a quantitative reverse transcriptase PCR. And lastly, we're going to be doing some protein expression assays by the means of ELISA. Okay, now I'm going to hand it over to Megan, who's going to talk to you a little bit about our cell migration assays.
Thanks, Eddie. So the purpose, another aspect of our experiment is to observe the migration of the cell's population, and we will be observing how far they're migrating after 3 and 6 hours after stimulation. Before testing, we will be first 3 wells will be experimental wells, which will have cells that are stimulated, and the next 3 wells will be control wells, which will have cells that are not stimulated. You can see that the cells will be scraped into a 1 centimeter wide line down the middle, and on the left and right sides, there will be no cells because we have scraped them off. The reason why we're scraping our cells on the left and right sides is so that it simulates a wound site where the cells will be migrating to depending on the current of flow. In our project, the flow will be from right to left, so we will be observing how the cells are migrating from the right side to the left side. The second part that we will be performing in our migration imaging will be to take 3 images along each side of the cells that have been scraped. So there will be 3 images taken on the left side and 3 images taken on the right side. The reason why we're taking 3 images on the left and the right is to ensure fairness so that you have different locations being taken pictures of and so that there's no bias towards one location. We will be measuring the distance from the starting position to the end of the cell population using the software ImageJ, which measures pixel distances and area values, so we're able to calculate the distance that the cells have migrated from the beginning position to their ending position. And after we have calculated the average values of the migration distances, we will be able to calculate a total average distance value for how far the cells have migrated. As you can see on your right-hand side, on your left-hand side, you can see an orange picture, and on the left side, you can see that we have scraped off all the cells, and on the right side, you can see the line where the cells have been scraped off of. And on your right-hand side, this is a chart of our migration data. Looking at this chart, you can see that the experimental cells have migrated much further than the control cells due to the electrical stimulation, which signifies that our experiment has been successful since the control has moved less than the experimental cells that were stimulated. Currently, we are finishing up our experimental testing for our specific aim 1, which is testing our linear device and the effects that it has on cell proliferation and migration. Next semester, we will be continuing with our specific aim 2, which is testing our radial device and its effects on cell proliferation and migration as well. We will also be doing an ELISA test, which is called an enzyme-linked immunosorbent assay, and we will be using this to measure the effect of electrical stimulation at the protein level to see how the stimulation is affecting the expression of the growth factors VEGF and BFGF. And if we have time, we will also be testing the most efficient device, either linear or radial, and we will be testing this in vivo on diabetic rat models that have been induced with a wound, which shows that it simulates a diabetic ulcer. And finally, here is some advice that we have for the future freshmen and sophomores. Coming from a large team, we have 13 people divided into 4 subgroups: the electrical subgroup, cell migration subgroup, cell analysis subgroup, and cell culture subgroup. We have a lot of different people working on different aspects of our projects all the time. So 2 of the main challenges that we've had so far are having a good method of communication within our team and also being able to divide an even workload amongst all our team members. However, being a part of a large team also has its advantages because if you have a lot of people on your team working on different tasks, it's really easy to be able to balance out your workload. If some people are working in the lab a lot, other people might be doing the cell analysis or data collection. And speaking of data collection, another issue that has come up in our team is due to the fact that we run tests every single week, we always have new data coming in every week, and we need to be able to keep up with our data organization so that we don't fall behind. And lastly, some Gemstone tips for success are to find a mentor who's involved in research related to your topic and has resources available. For example, we are really fortunate to have Dr. Fisher as our mentor because he's given us a lot of lab space and he has really great grad students who help us out in the lab. And another thing is to be flexible with your schedule because plans might change unexpectedly and to apply for grants. And also remember to make friends and have fun. Concluding remarks, we would like to thank Dr. Fisher, our mentor, engineering professor, Mr. Miller, our librarian, our lab graduate students, Kim, Bao, Martha, and Xiaoxing, and our demonstrator directors and staff, Dr. Cole, Dr. Scandal, Ms. Creek, Dr. Wallace, and Dr. Jones. And if you'd like to find out more information about our team, you can